So, very good evening, everyone. I am Dr.satyabrata, MDDM clinical pharmacologist, and today I am going to discuss on good clinical practice and pre-marketing trial phases. So, today's session learning objectives will be to understand the definition of good clinical practice, to know the evolution of GCP, that is good clinical practice, to explain the purpose of good clinical practice, to elucidate the principles of good clinical practice, to get the picture of pre-marketing trial phases. Then come to what is GCP? That is GCP. That is not good clinical, that is good clinical research practice. So coming to definition of GCP, according to ICH E6 1.24, good clinical practice is a standard for the design, conduct, performance, monitoring, auditing, recording, analysis, and reporting of clinical trials that provides assurance that the data and reported results are credible and accurate, and that the rights, integrity, and confidentiality of trial subjects are protected. Coming to WHO 1995 definition of good clinical practice, it is a standard for clinical studies which encompasses the design, conduct, monitoring, termination, audit, analysis, reporting, and documentation of the studies, and which ensures that the studies are scientifically and ethically sound, and that the clinical properties of the pharmaceutical product, diagnostic, therapeutic, or prophylactic under investigation are properly documented. Then there are some examples of atrocities in the past, that is Nazi war crimes, Tuskegee syphilis trial, thalidomide disaster, Jewish chronic disease trial, the Willowbrook study, the Milgram study, for which there is development of the GCP guideline. Then coming to evolution of ethical principles. In 1949, Nuremberg Code was evolved, and 1964 Declaration of Helsinki, and 1979 the Belmont Report, and Council for International Organization of Medical Sciences SIOMS in 1993. Then coming to Belmont Report 1979, respect for persons, that is autonomy of individuals, special protection for those with diminished autonomy, beneficence, maximize the possible benefits, minimize possible harm, justice, subjects should be selected fairly to ensure that the benefits and burdens of medical research are evenly distributed. Then cardinal principles, that is autonomy means that each person should be given the respect, time, and opportunity necessary to make his or her own decisions. Then beneficence obligates the researcher to secure the well-being of all study participants. Belmont Report 1979 maximize possible benefits and minimize possible harms. Then justice is a difficult and complex ethical issue. Attempts should be made to distribute the risks and benefits fairly and without bias. Who should bear the risk of a study and who should receive its benefits? Participants should not be selected due to class, socioeconomic status, or race unless justified by study objectives. Then in 1990s, the decade of harmonization. Then ICH, International Conference on Harmonization, International Conference on Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use. Primary participants: USA, European Union, Japan, regulatory and industry representatives, observers, Canada, Australia, Nordic countries, WHO as facilitator. GCP purpose: quality data plus ethics is equal to GCP. Data and reported results are credible and accurate, that is equal to quality data. Rights, safety, rights, integrity and confidentiality of trial subjects are protected, that is equal to ethics. Then the 13 principles of GCP: good clinical practice, ethical conduct, risk benefit assessment, subjects over science and society, adequacy of pre-existing information, scientific validity, independent review, medically qualified care, trained and experienced personnel, informed consent, efficiency of information handling, confidentiality, drug accountability, quality through systems and procedures. The GCP Principle 1 Ethical Conduct. Clinical trials to be conducted in accordance with the ethical principles that have their origin in the Declaration of Helsinki and that are consistent with GCP good clinical practice and the applicable regulatory requirements. The GCP Principle 2 Risk Benefit Assessment. Before initiating a trial, weigh foreseeable risks and inconveniences against the anticipated benefit for the individual trial subject and society. Initiate and continue only if the anticipated benefits justify the risks. Then the GCP Principle 3 Subjects over Science and Society. The rights, safety, and well being of the trial subjects are the most important considerations and should prevail over interests of science and society. Then the GCP Principle 4 Adequacy of pre-existing information. The available non-clinical and clinical information on an investigational product should be adequate to support the proposed clinical trial. Then GCP Principle 5 Scientific validity. Clinical trials should be scientifically sound and described in a clear and detailed protocol. Then GCP Principle 6 Independent review. A trial should be conducted in compliance with the protocol that has received prior institutional review board IRB Independent Ethics Committee IEC approval favorable opinion. Then GCP Principle 7 Medically qualified care. The medical care given to and medical decisions made on behalf of subjects should always be the responsibility of a qualified physician or when appropriate of a qualified dentist. Then GCP Principle 8 Trained and experienced personnel. Each individual involved in conducting a trial should be qualified by education, training and experience to perform his or her respective tasks. Then GCP Principle 9 Informed consent. Freely given informed consent should be obtained from every subject prior to clinical trial participation. Then GCP Principle 10 Efficiency of information handling. All clinical trial information should be recorded, handled and stored in a way that allows its accurate reporting, interpretation and verification. This principle applies to all records referenced in this guideline irrespective of the type of media used. Then GCP Principle 11 Confidentiality. The confidentiality of records that could identify subjects should be protected, respecting the privacy and confidentiality rules in accordance with the applicable regulatory requirements. Then GCP Principle 12 Drug Accountability. Investigational products should be manufactured, handled and stored in accordance with applicable good manufacturing practice GMP. They should be used in accordance with the approved protocol. Then GCP Principle 13 Quality through systems and procedures. Systems with procedures that assure the quality of every aspect of the trial should be implemented. Aspects of the trial that are essential to ensure human subject protection and reliability of trial results should be the focus of such systems. Do unto others what you would like others to do unto you. This is in Bengali. Then pre-marketing trial phases. So algorithm for classification of types of clinical research. Did investigator assign exposure? If yes, then it is experimental study. If no, then it is observational study. And in case of experimental study, random allocation. Is there random allocation? If yes, then it is randomized control trial. If no, then it is non-randomized control trial. Then in case of observational study, ask the question, is there any comparison group? If yes, then analytical study. Then analytical, if no, that is descriptive study. In case of analytical study, ask question, that is there any direction? If the direction is from exposure to outcome, then it is cohort study. If the direction is from outcome to exposure, then it is case control study. If exposure and outcome are the same time, then it is cross-sectional study. Then coming to clinical trial defined in NDCTR 2019, New Drug Clinical Trial Rule 2019 refers to any systematic study of a new drug or an IND, investigational new drug in humans to generate data for discovering or verifying its clinical, pharmacological, including pharmacodynamic, PD, PK, pharmacokinetic or adverse effects with the objective of determining its safety, efficacy or tolerability. Then this is the drug discovery and development process. Discovery and then candidate selection, preclinical, then clinical phase 1 early development, that is clinical phase 1, clinical phase 2A, clinical phase 2B, and clinical phase 3. Clinical phase 3 and clinical submission, that is phase 4, are late development. From preclinical to clinical phase 2B, it is early development phase. So drug discovery and preclinical development. First, target identification will validation D0, assay development D1, hit finding and hit to lead D2, then lead optimization D3, then in D3 there is lead optimization, after that candidate selection, then preclinical study. Then this is the discovery funnel. First, high throughput screening to find out 1000 of compounds that hits, and hit to lead, that is hundreds of compounds compared to hit to lead. Then lead optimization, dozens of compounds. Then candidate seeking 1 to 3 compounds, that will go to preclinical development. So preclinical proof of principle at lead optimization, dozens of compounds, and candidate seeking 1 to 3 compounds. In between that is preclinical proof of principle. Then after preclinical development, clinical development, that is phase 1, phase 2, phase 3. After that, new drug application will be apply, and after verification by the regulatory authority, the drug to be accepted if it is safe and effective according to the protocol. If it is not safe, not efficient, effective, then it will not be accepted by the regulatory authority and not to be come to the market. Then come to types of preclinical study. Preclinical studies, PKPD, that is pharmacodynamic studies, that is pharmacokinetics, ADME, absorption, distribution, metabolism, excretion, tissue distribution, mechanistic studies. Preclinical safety studies, safety pharmacology, and toxicology. Toxicology, there is general toxicology, genetic toxicology, other toxicity studies. Then coming to phases of clinical trial. Phase 1, safety, 20 to 100 participants to be taken, and generally healthy volunteer, but in case of HIV and cancer you can take patient. Then before that, IND, investigational new drug application, approved for testing in human. After preclinical state, investigational new drug application, that should be approved for testing in human. Then phase 1 started, then it goes through phase 2, safety and dosing. Dose finding should be done in phase 2. 200 to 400 participants to be enrolled in phase 2. Then in phase 3 there will be, it is therapeutic confirmatory. Phase 2 is therapeutic exploratory phase. Phase 3 is therapeutic confirmatory, confirm the safety and efficacy of this new drug, of this investigational product. And in phase 3, 300 to 5000 or more participants, patients to be taken, then or participants to be taken. NDA, new drug application, to be submitted after phase 3 to DCGI in India, Drug Control General of India, or other regulatory, if the trial is going in foreign country, then to other regulatory bodies of the respective country. Then after review of the regulatory authority, then phase 4 will be done, post approval surveillance. Thousands of participants will be taken for the post approval surveillance. Before that, after review, drug is approved, then it will come to phase 4 study. Then what is clinical pharmacology plan? Clinical pharmacology plan includes safety oblique PK, pharmacodynamic mechanism of action MOA, that is SAD, single ascending dose, MAD, multiple ascending POC, POM, mechanism of action, PGX, pharmacogenetics, TQT, thorough QT study, biopharm, food, BABE, biology, bioequivalence study, line extension, ADME, absorption distribution, metabolism excretion. Then special populations, renal, hepatic, pediatrics, age oblique gender, patient population. Then drug interactions, DDI1, DDI2, DDI3, DDI4, DDI5. So in phase 1, there will be single ascending dose, multiple ascending dose, and drug interactions 1, food effect, relative bioavailability, safety, and ADME in biopharm. And in phase 2, there is drug DDI2 and DDI3, DDI. Then in phase 3, special population, ECG, BE, bioequivalence, special population. Then phase 4, special population, DDI4. So this is a complex target identification, target validation, and hit identification. From that, lead discovery, lead selection, lead optimization, new chemical entity, preclinical trial, then investigational product, then clinical trial, then new drug. NDA apply after clinical trial in phase 3, then if approved by the regulator, then new drug. And after preclinical trial, IND, investigational new drug, will be applied. Then investigational product, we will call investigational product and undergo clinical trial. Then new drug application apply in the clinical trial after clinical trial phase 3, and then if drug is approved by regulatory authority, then new drug will be come to the market. And for target identification, lead identification, there are lots of things: random screening, serendipity, natural products, rational drug designing, and prodrugs, single enantiomer, biotechnology, molecular modeling, combinational chemistry, and core therapy. These are a lot to find out the target identification, and from the target validation, hit identification, then at last new drug will be formed through these steps. Then this is the book written by my senior Dr. Shogat Sarkar and his and co, and this is the postgraduate pharmacology book, and it is a very good book. Anyone can go through this book and you will get a lot of knowledge from this book. You can improve your knowledge if you go through this book. Also, if you want to gain more knowledge, then you can go through guidelines and others articles also. As ethics committee member, will you allow this study? This is a study development of a simple radiant heat induced experimental pain model for evaluation of analgesic in normal healthy human volunteers. Indian Journal of Pharmacology 2011. So you can go through these articles. You can find out the answer also. So is something in Bengali slok. I don't want to go through this, and that's all for today. Thank you very much for watching my videos and subscribing my channel. Thanks again. Jai Jagannath.